Okay, I think we can start. We have a lot of materials to go through, so we'll start right now. Thanks for joining us during your busy summer schedule. This is our first public seminar for bioinformatics since the establishment of the core. So my name is Junzi Liu. I'm the director of the Bioinformatics core, and joining me today are Dr. Olex and Mr. Chu. Both of them have been instrumental in establishing the single-cell sequencing data analysis pipeline in the core and have been involved in a number of single-cell data projects at VCU. So next. Here is the outline of today's talk. I will give a brief overview of the services provided at the core, and while waiting for people to join in. And then Amy and Shifeng will take over to discuss single-cell sequencing and the step-by-step illustration of the analysis pipeline. And remember, at the end, we'll have a live demo of the Loupe browser for interactive visualization. If you have any questions, you're welcome to send text in the chat, and we'll try to answer it through chat or at the end of the presentation. I also want to let you know that we'll have a 4-hour workshop on IPA Ingenuity pathway analysis scheduled on September 14th. It will be an in-person with 2 workshops, 2 hours for beginners and 2 hours for advanced users. So please stay tuned if you're interested in participating in that. Okay. Next. So what does Bioinformatics core do? Pretty much what we do is to turn your data on a hard drive or a cloud drive into a number of pretty pictures for data interpretation purpose. That's in short description. So in general, our core services center around omics data analysis. I'll talk in the next slides about what kind of data we're working on. And also, we can help on how to associate omics data with clinical data, how to mine public genomic data resources. If you have data sets that's not very standard or something very unique, we can also develop customized bioinformatics solutions. In general, we'll have support for grant application to support the entire life cycle of your grants. In the area of bioinformatics, we'll help with manuscript writing as well. We're also happy to train your postdoc on the data we've been delivered or the result being delivered, or provide consultation on bioinformatics solutions. We currently, the request can be received by sending emails to me or bioinfo@vcuhealth.org. We're working on the customer portal, but it should be ready for you to submit a request through that soon. Okay, go ahead. So for omics data analysis, most of our data requests actually are in the area of transcriptomics data, which typically involve RNA-seq, single-cell RNA-seq. And also in the area of genomics, we have done analysis on whole genome, exome, whole exome data analysis. We have done, we have pipelines for ChIP-seq data. to look at transcription patterns. We have worked closely with lipidomics core, metabolomics core, and proteomics core to conduct downstream data analysis to develop prediction modeling for phenotype prediction, disease prediction, and also to identify biomarkers. We have routinely performed pathway analysis, ontology enrichment analysis. So this is those are 4 big areas of omics data analysis service we provide. Next, please. So just to give you a brief idea of when and how to involve bioinformatics during your NGS workflow. In general, for sequencing, there are 3 big steps within the entire workflow. So you start with experimental design, and then you do the library prep and sequencing, and at the end is the data analysis. Oftentimes, we receive the data at the end after you did the sequencing, but we would prefer to be involved much earlier in the experimental design, where we can help with power analysis to determine the size of the reads, the length of the reads, and the depths of the sequencing, so on and so forth, which can be very important for a specific biological problem you're trying to address. Okay, go on to the next. So bioinformatics core work is one of the 3 quantitative cores with different focus, and it works closely with 3 other omics cores. We do have direct data transfer between genomics core and us. So if you sequence data at genomics core, please and want us to analyze, we can arrange a direct transfer of the data to the HPRC. And we are supported by the high performance computing cluster. That's where we store and analyze the large amount of data. Next. So now I want to focus on single cell. So we all know that single cell has gained a lot of attention in the past 5 years. It is a breakthrough technology, allow biologists to discover remarkable diversity in cell function throughout the development progression of disease. It's offered a much higher resolution for us to understand the dynamics in gene regulation. It is being able to do this by simultaneous gene profiling in thousands or even millions of cells. So besides single cell RNA-seq, which is very popularly used right now, single cell multi-omics is also under active development. And there's a lot of integration method as well. So as we know that single cell data are orders of magnitude more bigger, at least than bulk RNA-seq, which we were more familiar with. And through my conversation with a lot of investigators, one hesitation to generate single cell data is that the data is so overwhelming, and how to analyze them and get information from it would be really hard. So I think the goal of the seminar today is to give you a brief understanding about what we do at the core regarding the data analysis. So hopefully this can ease your concern over the data analysis. So if you need to do single cell sequencing in your research, to advance your research, so we're here to help, okay? So next, to address the data analysis challenge, recently there has been tremendous efforts or research into the computational method on analyzing this data. And to date, we know that there was over 1,500 data analysis algorithm or tools has been developed. So it means it's an open research field, but at the same time, it's getting mature. And there are a number of pipelines that has been very popularly used and has been to support many different single cell applications and has been published. So let's go to the next slides. So our presentation today will focus on the in-house single-cell RNA-seq pipelines developed. So there are several unique features of our pipeline. So it's a pipeline that's blends two widely used single-cell RNA-seq pipelines. Okay, one of them is 10x. 10x Genomics' own pipeline. We like that mostly because of the interactive exploration tools, which is a Loupe browser. And then the other one is Seurat. This is one of the first, but it's also most widely used R-based single-cell analysis pipeline. It has a lot of robust and sophisticated data analysis methods in it. In the meantime, it incorporates a number of latest development methods as well. So typically we use either one of them for analysis, but if you use Seurat, it's mostly R. You don't have good visualization. So so we basically divide the whole thing into one pipeline, and we have a part of it, the most part of it for in-house heavy lifting analysis with R-based approach, and also linear space approach for data alignments. And we will deliver the parts that you can actively explore through files that's compatible for Loupe browser. Okay, so our focus today will be we'll give you a step-by-step illustration of what we do in each step of the pipeline. So next, I'm going to hand the presentation to Amy and Shifeng, who will give an introduction of sequencing and also the pipelines. As I said, if you have questions, please you can use chats where we'll at the end we'll answer the questions. Okay. All right.
Thank you, Jinza. Yeah, so next I'm going to give a high-level introduction to single-cell sequencing along with some important terminology that's useful for analyzing the data, and then give an overview and start off our discussion of our in-house analysis pipeline. So at a really high level, and I love this figure, bulk RNA-seq data essentially takes your sample and blends it all together before it does sequencing. So your result is really an average gene expression across all the different cell types in your sample, which masks that cellular heterogeneity. Single cell sequencing, however, is able to individually and uniquely tag each cell in your sample before it is sequenced. Then once we get the sequencing data back, we're able to tease out the gene expression levels for each individual cell, which allows you to actually analyze and utilize that cellular heterogeneity that may be in your organ or tissue or sample that you're analyzing. So there are some pros and cons to bulk versus single cell sequencing that you should take into account when you're doing your experimental design. Single cell sequencing is more expensive than bulk RNA-seq. It's also more labor intensive and more complex statistical analysis. However, on the positive side, there's more detailed information of individual cells. And it also can reveal cellular heterogeneity and allow you to explore the subpopulations within your sample. Whereas bulk RNA-seq actually masks this and gives you that average expression across cells. So the bioinformatics core is able to actually help reduce some of the burden on the informatics side of things, as we're able to do all of the heavy bioinformatics analysis. So here's just some terminology that goes along with the 10x genomics pipeline, as well as sequencing in general. So with 10x genomics, they utilize barcoded gel beads, where each barcode is a unique identifier that tags a single cell. So these barcoded gel beads are associated with single cells and then emerged in an emulsion mixer. So each of these little spots is called a gem or a gel bead in emulsion, and that generally identifies a single cell. So each of these are, within each of these gems, the RNA molecules are tagged both with the unique cell barcode as well as a unique molecular index or UMI code. The unique molecular index tags each individual RNA molecule with its own identifier. So we're able to actually utilize that in our QC and analysis downstream. Once all of this tagging is done, all of the RNA molecules are pulled together and run through a traditional sequencer. So here we have our library, which is our pooled RNA molecules that have been tagged with both the UMI and the barcode. And these are run through the sequencer, and the sequencer outputs digital reads, which are stored in a FASTQ file. This is a kind of equivalent to having a bunch of puzzle pieces in a bag. So we know they came from a puzzle, but we're not really quite sure where yet. They're all mixed up. So each read has a read length in base pairs. That's just the number of base pairs that were sequenced. And then these reads need to go through a process called alignment to a reference genome. The reference genome can be equated to the picture on the jigsaw puzzle box, right? So that's your reference, and that's used in order to put all of these pieces back together to figure out where these reads came from on the genome. And after that is done, then we're able to count how many reads align to a certain spot on a genome, such as in a gene, and that is where we get our gene expression numbers from. So now that we've went over a little bit of terminology with sequencing, I'm gonna start off discussing our in-house analysis pipeline. So the very first thing that we always do with any sequencing data, single cell or bulk, or a whole genome or whatever, is we do the sequencing level quality control. This is done with the tools FastQC or MultiQC. And we always do this analysis before we move downstream to ensure that the sequencing is of good quality. However, we're not going to be going into the output of that tool in this presentation. Instead, we're going to start with the alignment step and alignment level quality control, which is performed with the 10x CellRanger software. That is followed by cell level quality control, along with sample integration and downstream analyses, which are all done in R using various R packages, but mainly we utilize the Seurat package. And finally, once we've done our analysis, we've developed a custom pipeline that can take the results from our R analyses and port them back into the 10x pipeline so that they can be visualized in the Loupe cell browser, which allows our investigators to interactively explore the results of our analysis. So back up to alignment, 10x CellRanger software can actually do there's a lot of functionality that it can do, but we primarily use it for alignment to a reference genome and assessing the alignment level quality control. 10x offers 3 ready-to-use reference genomes, the human genome, the mouse genome, and a merged human-mouse genome. So some might ask, why do we want to use a merged human-mouse genome? Well, some of the data that we analyze in the core is actually derived from patient-derived xenografts, where human tumor cells were engrafted into mouse and then utilized in experiments. And so then these tumors in the mouse are extracted and sequenced. So inevitably, you're going to get both human cells as well as mouse cells. What CellRanger is able to do is it is able to digitally sort and align reads to either the human genome or the mouse genome, and then identifies and tells us which gem or which cell, which species that's from. So we're able on the bioinformatics side to filter out all the mouse cells if all we're interested in is analyzing human gene expression. 10x also allows you to build a custom reference genome in case you need to align to a species other than human or mouse. And an example of how this can be used is one of our projects, we were looking at the microenvironment around a brain tumor in a mouse model. And in this particular mouse model, the tumor expressed a firefly luciferase gene, which is not native to the mouse genome. And so we wanted to get rid of these tumor cells because we only wanted to look at the mouse microenvironment around the tumor. So what we did is we created a custom reference genome. We added this one gene sequence to the mouse genome, and then aligned our sample to that custom reference genome. And then we were able to identify which cells actually express the firefly luciferase gene, and we could remove those easily from downstream analyses. And this plot that you're seeing here, you'll see several more later in the presentation. It's called a TSNE plot, where each dot represents an individual cell, and this plot in particular is colored by expression of this firefly gene. So the next thing after we do alignment is we want to do the alignment level quality control. So was the alignment good? And so CellRanger exports an HTML file with various amounts of statistics on it. And I'm just showing a screenshot of some examples here. So one of the first things we look at is the estimated number of cells. In this experiment, we were targeting 5,000 cells, and we ended up with 3,800, which is about expected and okay. We never get exactly 5,000, but it's the right magnitude. We also want to have greater than 20,000 reads per cell, because in order to do a robust differential expression analysis. So we're good on that. Another plot that I like to look at is the barcode rank plot. What this plot shows is all of our barcodes. So these are our gems or individual cells, and they're ranked by the number of unique molecules, or that UMI count in the cell. And so what 10x is able to do is able to look at this and look at how many unique molecules are in each of these cells, and distinguish the viable cells from those that are background, or from the empty gems that actually didn't get associated with the cell. And then in good barcode rank plots, you want to see this nice knee. And on the next slide, I have a few examples, a few different examples of this. And then two other plots that we can look at are, again, the TSNE projection. That's actually where each cell is colored by its UMI count. So you can see these cells here have a low number of unique molecules, whereas these have more. And then finally, because we aligned this sample to the merged human and mouse genome, we get this cell UMI counts plot, where we have our human cells plotted on the x-axis and our mouse cells on the y-axis. And then we have a bunch of cells kind of in the middle that are classified as multiplets. What a multiplet is when the CellRanger software says, oh, there's reads in here that align both to human and mouse relatively equally, so we can't make a determination of if this is a human cell or mouse cell. And so these cells are usually removed in downstream analyses. So a quick example of some lower quality examples, just to give a comparison. In this experiment, we were again targeting 5,000 cells, and we ended up with over 17,000. Big red flag there. Also, the mean reads per cell is much less than 20, so that's also an issue. When we look at our TSNE plot, you can see how different it looks from the previous one. So this is not a good TSNE plot. And then finally, again, this was also aligned to that merged genome, and it's saying all of the cells are multiplets, which is another big red flag. So it turns out there was actually some issue in the library prep prior to sequencing with this sample. And so working with the 10x help folks, we were able to figure out what that was and fix it when we reran this sample. And then finally, just to give you an idea of how different these barcode rank plots can look. So we have a nice me here, but all of our other statistics are way off. So this is usually what a well-defined me looks with a gentle slope here. In this sample up here, we're actually missing that well-defined me. It just kind of gently slopes down. So we initially thought this might be a problem. However, working with the 10x folks, we deemed that this was actually an okay sample to use. So we did continue to utilize this sample just with some more strict filtering criteria. Working with the 10x support is actually very helpful. In some cases, they're very knowledgeable and very helpful. All right, so once we do alignment, then we move down to doing the cell level quality control. And this is where we move from the 10x CellRanger software into the R platform. And this analysis is done with the Seurat package. So there's three main metrics that we focus on: N feature, which is the number of genes detected in each cell; N count, which is the total number of molecules detected in each cell. Again, that's that UMI count metric that we mentioned before. And then also the percent of mitochondrial gene expression, where a high percent of mitochondrial gene expression generally indicates that the cell may be dead or is in the process of dying. So we want to exclude those from our downstream analyses because it could skew our results. And these metrics are usually viewed with violin plots, as shown here, where again, each dot represents an individual cell and your y-axes are the count or the percent of that metric. So this is what a plot looks like before filtering. And we can see here that we have a lot of cells that actually have either no or very low number of genes. These are probably empty gems that the CellRanger software just did a cutoff and didn't quite catch these as being empty gems. So we need to remove those. Next, we can look at N count. And here we're actually looking for high outliers. So any cells that have really high UMI counts probably indicate that there may be two cells in that gem. So maybe two human cells in this case. And so we're going to want to remove those as well because they could certainly skew our results downstream. And then finally, and this is probably the one that we put the heaviest weight on, is the percent of mitochondrial expression. Generally, we see a population of cells below 25%. These are our good and viable cells that we continue with downstream. And then there's usually a population up around 100%. These are our dead or dying cells with a bunch that fall in between. So once we apply these cutoffs, we can regenerate our violin plots and see that in each plot, we now have a single population of cells that we can use in downstream analyses. And then here's just a few other sample violin plots just to give you an idea of the variety that you can see depending on your sample and tissue type. So all three of these here are actually good samples. We see two distinct populations, less than 25% and up around 100% in all of these. So these are very clean samples. However, these two samples over here are a little more questionable. If we look at the percent MT, we certainly do have a population around 25%, but then there's a lot more cells that have higher mitochondrial expression here. In the end, we decided we did use these samples. But we had to do some major filtering and in some cases had to remove about half of the cells from our analysis with this 25% cutoff here. This is actually just an example of how single cell sequencing data can help salvage samples. If this was a bulk sample, for example, then all of this gene expression from both the healthy cells and the dead cells would be merged together and we wouldn't know any different. It'd be hard to be able to tell that that was a bad run. However, with single cell data, we're able to get that fine-grained view of the cell quality and extract only the good cells for downstream analysis. Finally, in my last example, this is an example of an experiment that we discarded. We don't use this data any longer because there is no distinct population less than 25%. Everything is just scattered throughout with the mitochondrial expression. So this is just an example of something where we'd say this whole sample is probably bad. We're not going to use it. And now I'm going to pass it off to Zhufeng, who's going to talk about some of the downstream analyses that we can do with this data.
Thank you, Amy. Here I'm going to talk about our integrated pipeline for single-cell data analysis. So first, we will firstly go over the dimensionality reduction method before diving into more single-cell specific method. So dimensionality reduction is a transformation of data from high-dimensional space into low-dimensional space, so while retaining the meaningful property of the original data. As you can see from the big table with blue and green color, it illustrates the gene count matrix of one experiment. The row stands for cell and the column for gene feature. So the number of cells from one experiment can be something from 1,000 to 10,000, and the number of gene can range from 15,000 to 30,000. So the matrix can be somehow very large. Also, the high-dimensional data can be often redundant and time-consuming for computational during downstream computation during downstream analysis. With dimension reduction, so that we can transfer the high-dimensional gene expression profile to a low-dimension representation. In the meanwhile, retaining the relative distance between all the cells. So in this example, we have a low-dimension representation with 30 components. Afterwards, we are able to use it to do clustering without any computational issue. So the most frequent used method for dimension reduction for single cell are PCA, UMAP, and t-SNE. So PCA is a linear method, and the UMAP and the t-SNE are the nonlinear method. The results of dimensional reduction can be visualized by a UMAP plot. UMAP plot. In this case, the cells are plotted against top two most significant components. Here, each dot is a cell, and cells that are closely projected indicate a similar gene expression profile. Next. So clustering is actually the centerpiece of the single cell data analysis. Pretty much all the downstream analysis counts on the reliability of clustering result. So what is clustering in single cell? Clustering is basically to identify group of cells with similar gene expression. So commonly used methods for clustering are k-means, Loupe, and Leiden. So k-mean is the basic approach for clustering. On the other hand, Loupe and Leiden are the modularity-based, or you can call it community-based algorithm, which were specifically designed for single-cell clustering. So when we do clustering, there is an important question that is, how many clusters do we need, or how many cell population do we want? So for k-mean, we can simply predefine a parameter. For example, a cluster equal 5. Then the k-mean algorithm will return 5 clusters for you. But for Loupe and Leiden, they introduce a different parameter called resolution. So the resolution is a parameter to control the number of clusters. As we can see from the figure below, as the resolution increases, more and more clusters are emerged. There's something we need to pay attention to, because we don't want either under clustering or over clustering. Next. Next, we are going to talk about the integration of multi-samples. So what is integration in single cell, and why do we need integration? So Basically, in single cell, we want to align different sample in the same space. For example, in figure A, we have 3 different sample, wild type, heterozygous, and null. They are integrated and visualized in the same 2D space. So here comes the question. Why do we integrate sample together instead of analyzing them individually? So as we can see from the figure C and D, they are the UMAP plot for wild type and heterozygous. If we analyze them individually, so as you can see from the UMAP plot, the scale of the coordinates axis are very different, as well as the shape of the cell population. Therefore, the distribution of cell population are very difficult to compare if the sample, being analyzed individually. So on the contrary, when you do the integration, as you can see from the figure B, it's quite easy to identify similarity and difference across conditions. Also, the annotation are quite consistent across sample, so that you don't have to, annotate them multiple times. Next. So during the process of integration, we must perform in-sample normalization in order to remove the influence of technical effect. Also, in the meanwhile, we want to preserve the true biological variation. So in Seurat, they provide 2 methods for normalization, which are log normalization and SCT transformation. Log normalization is as a standard approach in single cell is well used in multiple toolkits. So you can refer to the formula that how log normalization works. On the other hand, SC transform apply the Pearson residue to remove the inherent dependence between a cell's average expression and the cell-to-cell variation. Next. So once we have the clustering results, we can move forward to the next step, which is the cell type identification. So this is the step when we need a lot of input from the biologists to determine the identity of each cell population. So there are several ways that we can manage to do the annotation. Firstly, we can compare each cluster against the rest of the cluster and acquire a set of highly expressed genes that are upregulated in this cluster. And the result can be present by a heat map. For example, in cluster 0, we have 10 genes, which are SOD2, MGP, and etc., which are highly expressed in cluster 0, comparing to the rest of the other cluster. Therefore, we can have the domain experts to determine that which cell type the cluster 0 is. Next, please. Secondly is the marker-based method. So in this case, biologists often know what cell population might be in the sample, either based on the existing knowledge or publication, but they just can't predict which cluster or which cell type. So if you can provide the cell type specific gene marker, and we can generate a dot plot based on it. So dot plot is a matrix present by dot. It basically shows the presence of marker in current cluster with dots in different sizes or shapes. A drawback of a marker-based method is that they wouldn't cover the novel cell type. Therefore, it would be better to take advantage of the both above mentioned method together. Next, please. There are some other methods that can automatically predict cell type of cluster instead of manual annotation. This method takes advantage of machine learning or deep learning technique, which require a large amount of training data. And the accuracy of this method really, relies on the quality of the training data, which is the whether or not the annotation is accurate. Next, please. Differential expression analysis. Differential expression analysis is a method to discover quantitative change in expression level between experimental group, so that some of the genes are upregulated and some downregulated. For example, in the comparison of sample A versus sample B, we can perform differential expression analysis on certain cell population. In this case, KRT8 positive progenitor cell population. Then we can acquire a list of differential expressed gene, aka DEGS. The way that we visualize this gene is we can create a volcano plot. Next week. Oh, thank you. Enrichment analysis. Gene set enrichment analysis, as known as GSEA, is to identify classes for differential expressed gene and to see if those genes were associated with any diseases. And pathway analysis is about querying the existing pathway database with differential expressed gene to identify pathway which are significantly impact in the given condition. So in our core, we mainly use the Ingenuity pathway analysis as known as IPA. So it is a commercial database for pathway analysis. It produces massive amount of information for each request. The figure below show the result of one pair of comparison, which is the KRT8 positive progenitor cell population between sample A and sample B. So as you can see, the blue bar in the plot indicate that these pathways were inhibited in the progenitor cell in terms of sample A versus sample B. And the orange bars indicates these pathways were activated in this cell in terms of the comparison. And white bar mean there is no clear signal for activation or inhibition. And green bar stand for that the information is, just insufficient to make a prediction. Next, please. So we have expanded the existing pathway analysis by merging the pathway analysis result from one individual cell population together into one table. So in this way, this allowed us to have a comparative view regarding, how different cell population functions. Next, please. Trajectory inference and pseudotime analysis. As we know, single-cell RNA sequencing offer a high-resolution snapshot of gene expression profile. So the snapshot can reflect a transient state of biological system. So trajectory inference and pseudotime analysis enable the study of dynamic change in gene expression so that we can infer cell lineage using computational method. The commonly used tool for trajectory inference and pseudotime analysis are a slingshot and monocle. As you can see from the figure, both trajectory inference and pseudotime analysis are based on the result of clustering, and both analyses require a starting cluster or a starting point. For example, stem cell. Trajectory inference define a temporal order in cluster level. As you can see, the connection between cluster are called lineage. Basically, if clusters are adjacent to each other, we consider the lineage between them are meaningful. Otherwise, if there are lineage between non-adjacent cluster, we reckon those lineage don't have any biological meaning. On the other hand, pseudotime analysis defines temporal order in cell level. It assigns each cell a pseudo timestamp to indicate the order of cell. This helps with understanding of how cell change state or how cell fate decisions are made. Next page. Thank you. Besides the figure and plot I've mentioned in my talk, there's some other plot we can adopt in order to visualize the single cell data. For example, volume plot, feature plot, and bar plot. So the volume plot is basically show the distribution of a gene and how genes express differently in different cluster or different sample or groups. Feature plots, that is very intuitive. It shows that in which part or which cluster the gene are highly expressed. And we have bar plots. It show how cell population change across sample. That's the end on my part. Amy.
Okay, thanks, Yufeng. So next we're going to do the Loupe browser live demo, but before we get into that, I just wanted to pause and see if anyone had any burning questions that you wanted to ask before we move on. I think most of the questions in the chat were answered. Okay. All right, so let's get into the demo. So I'm gonna just demonstrate the Loupe cell browser that is offered through 10x. This is free software. Anyone can download it from the 10x Genomic site. And I'm gonna be using the acute myeloid leukemia blood cell tutorial data that is made available within the browser. And we're gonna go through an interface overview, differential gene expression analysis, a manual cell type identification, and exploring substructure. All right, can everyone see my desktop? I just have to ask to make sure it's still sharing. Got a thumbs up, thanks, Chris. Okay, so to get to the tutorial data, and this is automatically loaded by default when you install the Loupe browser, you can just go up to help and hit load tutorial data. And so this is the main interface of the browser. I'm using Loupe version 6. I believe there's a newer version out at this point though. In the center here, you have your main window where your results are displayed. The default is a TSNE layout. Again, where each dot is an individual cell. And in this case, they're colored based off of the clustering that, that was done prior to this. So on your left, you have different zooming options, panning options. You can export the plot. You can also change the weight if the dots are too small. You can also change the weight of the dots to suit your preferences. Up here, we have a split on category, which we're going to use that functionality a little later. You have your pointer as well as some lasso and brush tools. Here is where our layout options are. So with this data, we only have a TSNE layout. Generally, there's also a UMAP layout. And then in any data that you get from our core, there are additional layouts, such as PCA layouts, so that you can visualize your data differently. And we can import any type of projection into this workspace. Then we have different annotation type tools. So right now we're on categories. There's also a gene and feature expression, which we'll show in a minute, as well as some others. And then below that are the specific options available for that particular annotation category. So right now we're coloring the cells by graph, the graph-based clustering. We can also color them based off of k-means-based clustering or the AML status. Down below at the bottom here, we have our significantly differentially expressed functions, as well as the, if I go back to graph-based, the differential expression results show in a table down here. You can also visualize these as a heat map. or as a violin plot for some analyses. Okay, so that's just an introduction of the layout of the interface. So the first thing we're going to do is do a differential gene expression analysis. There are two types of differential analyses that you can do. One is globally distinguishing, the other is locally distinguishing. What globally distinguishing does is showing the actual results that are showed. Hello? Sorry. Globally distinguishing is the results that are currently showed. So what it does is for each cluster, it takes that cluster. So here we have cluster 1. It takes this cluster and does a differential expression analysis compared to all other cells in the sample, regardless of what cluster they're in. It groups them all together to do that analysis. And it does that for each cluster. Locally distinguishing only does it for the selected clusters. So say we actually don't want to include a few of these clusters in our differential expression analysis. We can deselect them and then run a locally distinguishing analysis. And it will only give us the differential expression of, say, cluster 1 versus all the other clusters that are selected. So clusters 11 through 16, and you can see those results pop up here. For moving forward with our tutorial, we're actually going to do a differential expression analysis on the AML status annotation. For this particular analysis, locally and globally distinguishing analyses are the same because we only have 2 samples, 2 annotations for our data. So if we go ahead and run this analysis, here we see down here, we have our normal cells. And these are the significantly upregulated genes associated with our normal cells compared to our patient. And then we have the inverse, where we have the significantly upregulated cells in our patient sample compared to our normal. So that's the basics of how to run a differential gene expression analysis. And you can, if you need to export these results, you can click on this button and export them as a CSV file. So next, say we want to dive into this a little deeper, and we're interested in which cells actually express CXCR4. We know that it's mostly upregulated in the normal samples, but what exact cells express that? So if we click on this, we can add CXCR4 to what's called an active feature list. And here you'll see that the colors changed on the main screen, as well as our sidebar here. Now we're actually in the gene feature expression functionality. And this is our active feature list here. So we have CXCR4 listed. And down here, you can edit the color palette. If you don't like reds, you can do rainbow or another color palette. You can also filter on gene expression down here. Okay, so one thing, so these are blood cells. So in the tutorial, what they actually do is manually identify which cells are B cells specifically. And we do that by using biomarkers or specific genes that we know are upregulated in B cells. So let's go through that manual process of identifying which cluster of these cells are B cells. So what we want to do is we're going to use CD79A and CD79B as our biomarkers for B cells. So here's CD79A, and we're going to add that to our active list, and here's CD79B. And as you can see, primarily it's this set of cells right here that have expression of these genes with a smattering of cells elsewhere. So we want to select these cells and actually annotate them and give them a specific annotation. So what we're going to do is we're actually going to use this filter feature, and we're going to say any cell that has a read count of greater than 1 for either of these 2 genes, we want to filter those. So it automatically selects them. And so we're going to give this new category the name B cells, and then we're going to name this cluster. This is all B cells. Got a spell here. And then we hit save. And now you can see we've actually moved back to our categories functionality, and we have a new annotation called B cells. Okay, so that's great. We've identified all our B cells, but there are a few out here that we don't want to include in this group. So how do we get rid of those? If we go up to our split category and click on B cells, this actually does a split screen where we have all unassigned cells and all cells that are just in our B cell group. We can utilize the lasso tool to select all of these extra cells that we do not want in our group. We don't want them in our B cell category, and we click on unassigned from B cells. And they are gone. They are back into the unassigned category. So that's one great functionality of the split screen, and this enables you to actually tease out information like that. So the next thing we want to do is we kind of notice that this cluster here, these are all B cells, that's great, but there's some obvious substructure to this group, primarily three to four different clusters. So now we're going into the last part of the pipeline. We're going to explore this substructure and see if these actually have distinct gene expression profiles. To do that, we can use our lasso tool again and select each cluster. We want it in the B cell category. We're going to just call it a random cluster 1. And now you can see those cells were removed from the all B cells annotation and put in their own group. So we're going to do this again for the second one. Save. And then this is a little redundant because it's already in a cluster by itself, but just to demonstrate, click cluster 3. So now we have clusters 1, 2, and 3, and you'll notice that all B cells group went away because we reassigned all of those cells to a different cluster. Now we can actually do a differential expression analysis between these 3 clusters, but it's important to pick the locally distinguishing gene expression here because we're only interested in the contrast between these 3 clusters. We do not want to include any of the cells from the rest of the data set. So we choose locally distinguishing and hit our calculator button. And here we go. So here's our expression analysis. These are the top upregulated genes, most significantly upregulated genes. That's the default ordering for cluster 1. And then we can see we have a different gene set for cluster 2, and an even different set of genes that are significantly upregulated for cluster 3. So we can see there are possibly 3 different subtypes of B cells here in our data set. And so that actually concludes the live demo. There's a lot more functionality that you can do with Loupe browser. And again, you can export these results, and there's a recluster analysis that can be quite useful as well. But we're going to go ahead and end there. Hopefully that gave you a taste of what it can do and how helpful it can be towards exploring your data. And this presentation is being recorded, but I also have screenshots here just walking you through that exact tutorial. It's also up on the 10x website, and they also have additional tutorials as well. So that ends our seminar. So we want to thank everyone for joining, and we can take any questions.
Yeah, we have a lot of questions through the chat. One of the last questions I'm answering is from Danielle, who's asking if the haplocytes, the cell is too vulnerable for single cell. So I can't answer that question, yes or no. I think I would suggest a pilot study or to test if such cells will survive. In our past experience, we definitely see there are cells more alive, and there are other cells that's just very vulnerable, and we end up not getting good data. So also maybe talk to other investigators with similar experience and see what they say, if that helps.
Yeah, we can also talk to you can contact 10x support. They may actually have more of an idea on different cell types because they generate a lot of test sample data sets and work with way more people than we do. So they've been very super helpful in the past for different projects. If we run into questions or something just looks odd, they're more than happy to dive into the data, into the details, and help us figure it out. So
I think we previously we have several investigators have the same kind of data set. I think they have done the single nucleotide sequencing instead of single cell sequencing.
Yeah, but I don't know whether I think they're trying to eliminate the large cells. Okay. Oh, or they're trying to capture those. I can't answer that question. I think a good person to talk to is Dr. Seurat, who might be in the audience as well. I don't know whether he's still here. Dr. Seurat, do you want to say something? Anyway. All right. Yeah, Danielle, so if you want to reach out to Dr. Seurat, he could be very helpful. And Dr. Zhou, Dr. Huiping Zhou, who's also very experienced in this area. Any other questions?
Yes,
Go ahead.
Yeah, if like we have data from data sets and we want to analyze what we need to do, what we need to provide you exactly in order to help us run the analysis. Can you hear me?
Yes.
Yeah. So my question, what we need to do to provide exactly. Let's say that so I have a publication with a data set, okay, and I want to analyze a gene, okay, that I'm interested in. And I want to ask you for help. What I need to provide you exactly, please?
Oh, what you need to, if you can provide us the link to the data sets and the publication, we can help you to download the data and reanalyze the data for your own interest.
Yes.
Yeah.
Thank you.
Yeah. Any other questions? So if you have any feedback to the presentation, for example, request for another round of demo, things like that, we'll be happy to let us know, and we'll be happy to arrange that in the future as well. Okay. If I don't have any questions, thank you everyone for joining us. And the recording will be put online soon. We just need to figure out the place, and we'll let you know by email. All right. Thank you. Have a good day. Bye.
Thank you,
everyone.
Have a good day.